The ELISA is widely used in laboratories to detect and quantify the presence of specific substances, such as protein, peptides, antibodies, hormones and antigens, in samples. As a research tool that can quickly, conveniently, and accurately detect target proteins in samples. The ELISA kit has been widely used in many important research fields. Room temperature equilibration. Equilibrate the reagents at room temperature for at least 30 minutes. Preparing standard. Take 1 vial of standard out of the kit. Centrifuge at 6,000-10,000 RPM for 30 seconds. Dissolve with 1 milliliter of sample diluent. And aim the pipette tip at the bottom of the vial and pipette repeatedly for 5 times to aid dissolution. To avoid foaming, mix well to get standard S7, put aside. Take 7 1.5 milliliters centrifuge tubes in sequence. Add 250 ml of the sample diluent each. Aspirate 250 ml of S7 into the first centrifuge tube. Pipette and mix gently. Aspirate 250 ml from S6 into that second EP tube. Pipette and mix gently. And so on for standard ratio dilution. S0 is the sample diluent. Take out the plate, which is detachable strip. Prepare on demand. Remove the unused strips and put them into tin foil bags. Seal and store at 4 degrees Celsius for standby. Adding sample, setting standard well and sample well to be tested respectively. It is recommended to run duplicates for the standard and sample. Add 100 ml of standard or sample to be tested to each well. Cover adhesive strip and mark. Incubate at 37 degrees Celsius for 2 hours. After incubation, discard the adhesive strip and liquid, spin dry without washing. Add 100 ml of biotin labeled antibody working solution to each well. Cover with a new adhesive strip and mark. Incubate at 37 degrees Celsius for 1 hour. After incubation, discard the liquid and spin dry. Wash plate 3 times. 200 ml per well. Soak for 2 minutes each time. Spin dry. Add 100 ml of horseradish peroxidase labeled Avidin into each well. Cover with a new adhesive strip and mark. Incubate at 37 degrees Celsius for 1 hour. Discard liquid in the well and spin dry. Wash plate 5 times, 200 ml per well. Soak for 2 minutes each time. Spin dry. For the last wash, spin dry the plate and then flip it upside down and pat it on absorbent paper 3 to 5 times. Coloration. Add 90 ml of substrate to each well in sequence. Incubate at 37 degrees Celsius in the dark for 15 to 30 minutes for coloration. Termination. Add 50 ml of stop solution to each well in sequence. Terminate reaction. Read values within 5 minutes after reaction termination. Measure the optical density of each well sequentially at 450 nanometers using a microplate reader.